So now in today's lesson, where we're looking at photosynthesis, we will look at the structure of the organelles that are involved in different biochemical pathways that ultimately are involved in the conversion of water and carbon dioxide from the air to those organic molecules such as glucose. And this process is mediated with energy from the sun. And we call this process photosynthesis. And again, in today's lesson, you'll be introduced to the appropriate terminology, and you should be familiar with their proper use. And the terminology is photolysis, the Calvin cycle, the light and the dark reactions of photosynthesis, and cyclic and non-cyclic phosphorylation. You should be able to explain those chemical changes and energy conversions that are associated with photosynthesis, and you should be able to describe and illustrate those matter and energy transformation processes that occur during photosynthesis, and to be able to describe the role of chloroplast and chlorophyll in photosynthesis. So we are going to go through all this in great detail, and at the end I just have a video that you guys can watch that will really reinforce all of these things that we are going to talk about in our lesson in hopefully an entertaining way for you. And again, all of these material are explained in detail in your biology textbook that I have shared with you. You can go ahead and read the chapter on photosynthesis. And there's always really interesting animated YouTube videos that you could look at to get a better idea of the processes of photosynthesis. So whether directly or indirectly, photosynthesis provides nourishment to almost all living things on Earth. We have the autotrophs, or in other words, the self-feeders that are able to photosynthesize and are able to photosynthesize and produce those important organic molecules from their inorganic raw material, from starting material that they obtain from their environments. Under this category are plants and also some algae that are photoautotrophs, meaning these are organisms that use light as a source of energy to make their carbohydrates, lipids, proteins, and other organic molecules. Then you have heterotrophs, and heterotrophs obtain their organic material by eating plants and other animals. So heterotrophs, in a way you can appreciate, are completely dependent on the photoautotrophs, those plants and algae, for their food source and also for oxygen. And oxygen is a byproduct of photosynthesis that is produced by these organisms that carry out photosynthesis. And although if we look at the basic equation for photosynthesis, and this basic equation is very simple, and here would be the basic equation for photosynthesis, the actual step-by-step pathways that convert those reactants of carbon dioxide and water to the products, which is glucose and oxygen, are actually those step-by-step pathways are very detailed and very complex. But if we break it down to its really bare minimum, carbon dioxide, which the plants absorb from the atmosphere, along with water that they absorb from the soil, they interact using the energy from sunlight to produce glucose, oxygen, and also some water. And this can be represented by this word equation. So if you just read it out, carbon dioxide plus water plus light energy gives you glucose, oxygen, and some water. So if we look at the molecular formulas, then we can really summarize this photosynthesis pathways into this chemical equation You have carbon dioxide, H2O plus light energy gives you glucose, oxygen, and H2O. And if you balance this chemical equation, you will find that it will take 6 molecules of carbon dioxide, 12 molecules of water, and to produce 1 molecule of glucose, 6 O2, and 6 H2O. And just notice that in this true form, water is present on both sides of the equation. And even though 12 molecules of water are used, 6 molecules of water are produced at the end of this, at the end of photosynthesis. And so you can further simplify this equation to just be 6 CO2 plus 6 H2O plus light energy will give you 1 molecule of glucose and 6 molecules of O2. But just be aware that water is consumed and also water is produced at the end of photosynthesis, but the net result is the consumption of 6 H2O overall. Now let's look at the structures that are involved in photosynthesis. In plants, and here in this picture, and this picture shows a picture of a leaf representing a branch from a plant, from a maple tree maybe. Photosynthesis occurs within the chloroplast, and the chloroplast is a structure that is found within leaves. All of the green parts of these plants, including any green stems, any green unripened fruit, all of these green structures contain chloroplasts. But the leaves are where they are most concentrated and is the major site for finding chloroplasts. And just to give you some numbers, there are approximately 500,000 chloroplasts per square millimeter of the leaf structure. So if you were to zoom in and look very closely at a square millimeter of leaf structure, it would be packed with approximately 500,000 chloroplasts. And the majority of these chloroplasts will be contained in the mesophilic cells, which are this middle layer. So if you look at the cross section of a leaf, so imagine we have a leaf and we have sliced through it and we're looking at a cross section. The very top of a leaf is a layer called the cuticle, and then there's a layer of cells called the upper epidermis. In the middle of the leaf, there's the mesophyll, and underneath it, there is the lower epidermis. And within there are some air spaces. There is the stoma. This is an important structure. I'm not sure if you've learned about this in previous biology courses, but this structure opens and closes and controls how much water vapor exits and enters the actual cell. And in the middle, you'll have the vascular bundle and all the other structures. But within this middle mesophilic layer of cells, that's where the majority of the chloroplasts are found, within these cells that are present in the middle of it. And so if we look a little bit more, and if we look at inside the cells, we'll see that inside the cell are these structures called chloroplasts. So if I take you, actually, let me take you back to, where did we discuss, maybe it was actually in our previous unit, wasn't it? Let me just grab that image. Okay, so when we looked at, sorry if I'm making you guys nauseous by flipping quickly through these pictures, but remember this image of the plant cell. And within the plant cell, excuse me, within the plant cell, you had the chloroplasts. And these are the chloroplasts. And you can see they are, inside them is packed with these thylakoids. And when the thylakoids are arranged together in these stacks are called grana. So let's go back to this lesson's image. So here is another image of that chloroplast. Let's go in closely. So there's that chloroplast from within a plant cell. And in this picture, obviously, this is a very simplified picture. Just there's one or two of various organelles represented here. But if you can imagine in real life, there would be many more chloroplasts, many mitochondria, a lot more of this endoplasmic reticulum and all the other organelles. A single nucleus, but many more of these various other organelles, and certainly a lot more involved and a lot more complex. But in this simplified vision, you can see there is the chloroplast. And then if we take a closer look at the chloroplast, within the chloroplast also has a double membrane. We have the inner membrane and we have the outer membrane. And in between, there is this space, which we refer to as the intermembrane space, or the space in between the membranes. So in the chloroplast, you have this, this interior space is filled with a semi-liquid type of material, and this semi-liquid type of material within the inside of the cell, we call this space the stroma. And in there's these membrane-bound sacs. So these sacs are also have a membrane surrounding them, and these are called thylakoids. So each one of these is a thylakoid, and if you take a close look at it cross-section, you'll see that it also has a membrane, and you can see the lipid bilayer represented here in this image. And when these thylakoids are arranged, stacked on top of each other, these are called grana, or when it's a single one, we call it a granum. All of them together in the plural form, we call them grana. And these grana kind of look like a stack of pancakes or a stack of pitas or what have you. So these grana then are interconnected with one another with this membrane channel that bridges them, and these are called the lamellae. And that's where photosynthesis takes place within this chloroplast. It takes place partially within the stroma, within the space here, and partially within the membrane of the thylakoids. And if you look closely at the membrane of the thylakoids, you can see that it is dotted with these chlorophyll molecules that are embedded within the membrane of the thylakoids. Excuse me for one moment. Right. So within the membrane, there's these chlorophyll molecules, which help to absorb light energy. If you look closely at what a molecule of chlorophyll looks like, this here represents, this here is a chemical structure of a molecule of chlorophyll. I'm just gonna try and zoom in so we can see all of it, or rather zoom out. Let's look at all of it. So this would be the molecule of chloroplast, and as you can see, this would be the part which could be embedded within the bilayer. This part is a hydrophobic hydrocarbon chain that would fit between the lipid portion of the chain, so it would be embedded within here, and this would be the portion that would be more hydrophilic, and it would be closer to the top, to the surface of the bilayer. So as we said, photosynthesis takes place partly within the stroma and partly on the membrane structure. And the membrane structure contains not only these light gathering pigment molecules of chlorophyll, but also contains other proteins that mediate the electron transport chain that ultimately generate ATP. So this stack of photosynthetic structures then allows for a greater surface area within these chloroplasts, which means that you can have more photosynthetic structures that can be squeezed into this very small amount of space. So really, by stacking and squeezing and having all of these membranes all concentrated within here, you have a much greater surface area for photosynthesis, and more surface area means more reactions and more photosynthetic products. So these structures that are involved in photosynthesis, these molecules of chloroplast, these are pigment molecules. And chlorophyll is an example of a pigment molecule, and there's two types of it. There's chlorophyll A and chlorophyll B, and both of these are very structurally similar in that they pretty much have this similar structure. They have this hydrophobic long tail, and they also have this porphyrin ring here with a central magnesium atom that is surrounded by these hydrocarbon rings. And if you notice, you'll see that there's alternating double bond structure and then this long hydrocarbon hydrophobic tail. This porphyrin ring contains those electrons that would absorb light energy and kickstart the photosynthetic process. And the long hydrophobic tail then works like an anchor, holding the molecule within the membrane. And if you think about what the two types of chlorophyll A and chlorophyll B are, chlorophyll A and B are different in only one position, and that is that chlorophyll A contains a methyl group, CH3, while chlorophyll B contains an aldehyde group, a COH group. And these are just the two differences that affect what type of light and what wavelength of light this molecules A and B, chlorophyll A and B, can absorb. So when we think about plants, or more specifically chloroplasts within plants, within leaves, absorbing light energy. We're actually talking about a great range of energy, or all the wavelengths that are given off by the sun, which we refer to as the visible light. And this visible light is visible to the human eye. And although we see light as a clear or white light, it's actually composed of all the different wavelengths that we see as colors of the rainbow when it's passed through a spectrum and spread out. And you can see all the colors, namely red, orange, yellow, green, blue, and purple or violet. And each of these colors travels through space, carrying a certain amount of energy. Some of these wavelengths are better absorbed by plants than others. So if we take a close look, we see that various pigment molecules absorb maximally at different wavelengths. So if we look at, for example, chlorophyll A, which is the most important of all the pigments, and chlorophyll B also, these absorb light, A and B also here. So there's a portion of it that absorbs a bit in the red part of the spectrum. But if you look, the greatest relative absorption, which is plotted on the vertical axis of this graph here, you'll see that the highest absorption of chlorophyll A and B takes place in the blue violet region, and then a little bit in the red region. And the blue violet region is this region, around 400 to 500 nanometers, this area. And then a little bit in the red region, which is around the 600 to 700 nanometer range. And the wavelength in nanometers is shown on the horizontal axis. And the wavelength that they actually absorb the least is in the green range. If you see, there's not much absorption happening with chlorophyll A and B in the green range. So the chlorophyll A is this dark green line. And if we follow it, we'll see that in the green area, there's not much absorption. Same with chlorophyll B. If we look at it and we follow this light green trace, we'll see that in the green, they have the lowest absorption in the green region of the spectrum. And that's because this is the wavelength that tends to pass through the plants, or it gets reflected back. And this is why we see leaves as looking green, because this wavelength is not absorbed, passes through, and gets reflected back most of the time. So that's the wavelength that's left to be detected by our eyes. So if we only rely, if I say we, I mean the organism, the plant, if it only relies on chlorophyll A and B to absorb, well, chlorophyll A and B really absorb only in this region of the spectrum, mostly a little bit down in the red region. But all of these other wavelengths that are present in natural light, in sunlight, all of these wavelengths are wasted in a way. So instead of only relying on one type of pigment, plants can carry many different types of pigments that will absorb different types of wavelengths. These are called accessory pigments, and they are present not in the same amount as the chlorophyll A and B, but they are present in smaller amounts. And examples of these are the carotenoids and the xanthophylls. So for example, if we look at the carotenoids, we'll see that the carotenoids absorb a little bit further, shifted towards a little bit more towards the red end, a little bit more upstream here. And their least absorption comes here in the yellow and red range. So if we trace the carotenoid absorption, we'll see that it really drops off once you get to the yellow, orange, and red section. And that's why fruit and vegetables such as carrots that have a lot of carotenoids look orange, look red, yellow, or orange, because this is the part of the spectrum that is not absorbed by the plants that carry predominantly carotenoids as their pigments. And what happens is that during the growing season, where the leaves are growing and actively green, but once the growing season is over, these chlorophyll molecules tend to break down as the weather gets colder and changes. And it's really the presence now of these accessory pigments that help with photosynthesis. And that's why you start to see the colors yellow, orange, and red in the leaves at the end of growing season during fall. And again, these accessory pigments are the ones that help to absorb the remaining wavelengths that are present in sunlight, the wavelengths that are not used and absorbed by the chlorophylls. And so more specifically related to the process of photosynthesis, although only chlorophyll A can absorb the energy that is required to excite or activate the electrons in its pigment, in its molecule, these other pigments are also able to absorb energy at their own wavelengths and pass that activated electron on to chlorophyll A. And so in this way, the chloroplast is able to really absorb the full range of visible light. And this is referred to as an action spectrum. And altogether, these combined absorption abilities of all these different pigment molecules can be expressed and plotted in this action spectrum. And this might be different from one plant species to another, and also depending on the season as well. So there's some homework questions for you to think about and consider, try out. So now, again, looking closely at photosynthesis. So here we have the chloroplast molecule. Here is the stack of thylakoids. And when we looked at that simplified chemical equation for photosynthesis, it wasn't really obvious that the process the entire process of photosynthesis takes place really in two very distinct stages. And those distinct stages are the light-dependent stage. And by light-dependent, it means that it needs light energy. It is responsible for capturing this light energy from the sun and converting it into a form of chemical energy, while the second stage, the light-independent stage, takes that chemical energy and actually then produces glucose. So the light reaction takes place within the thylakoids, and the light-independent stage, or the Calvin cycle, takes place within the stroma of the chloroplast. So in the first stage, the light-dependent stage that takes place within the thylakoid membranes, again, you have multiple steps. You have step 1, which is the photoexcitation of the electrons. You have step 2, which is the cyclic electron pathway. You have step 3, which is the non-cyclic electron pathway. And then finally, you can move on to stage 2, which is the light independent stage, or the Calvin cycle. And again, within this cycle, the Calvin cycle, there's a number of steps as well. There's the carbon fixation step, there's the reduction of PGA, and the RUBP generation, and so on until finally you have produced glucose. So let's look at these closely in detail and slowly go through them step by step. So the first stage, the light-dependent stage, takes place within the thylakoid membranes. And as you remember from our picture up here, we said that within the membrane, you have these chlorophyll molecules that are embedded in the membrane through their long hydrophobic hydrocarbon tail. And it's this porphyrin ring here with a magnesium at the center that is responsible for the absorption of sunlight and the excitation of the electrons. So that's the photoexcitation stage. Let's take a closer look at that. So the photoexcitation of the electrons. So in the chloroplast, and here we are looking at the surface of the thylakoid membranes within a chloroplast, light is not just absorbed by independent pigment molecules, but instead by molecules that are associated with proteins in a cluster that we refer to as a photosystem. The photosystem has a number of chlorophyll molecules and some accessory pigments that are set all together in a protein matrix that's embedded in the thylakoid membrane. And altogether, the structure is referred to as the antennae complex, and the light-dependent reaction then begins when you have a photon of energy, a photon, a unit of light energy that gets absorbed by one of the molecules within this photosystem complex. And upon the absorption of this photon, there's a transfer of energy, and an electron gains this energy from the photon and gets raised to a higher potential energy level. It becomes excited. But prior to absorbing this energy, the electron, which is at its lowest possible energy level at the ground state, that's where it starts with. But when it absorbs that photon of energy, it's excited and goes from the ground state to the excited state. So then this electron then moves from molecule to molecule until it reaches a specific chlorophyll, a molecule called the primary electron acceptor. And then depending on the proteins that are associated with this chlorophyll A molecule, the photosystem can be one of two possible types. You can have a photosystem 1, which is also called P700. Because its chlorophyll A molecule absorbs most efficiently at 700 nanometers or red light. Or you can have photosystem 2, which is called the P680, because it absorbs best at 680 nanometers, also within the red light region. But regardless of which one of these photosystems captures the electron, the energy that's stored within that electron can then be used to ultimately produce ATP and other energy organic molecules, and it can go through two possible types of electron pathways. So here we have now the cyclic electron pathway, but there's also the non-cyclic electron pathway. So these are the two types of electron pathway that excited electron can go to once it's reached that primary electron acceptor. So here is the cyclic electron pathway. So the electron has been excited here. Here we have photosystem 1 or P700, and it has absorbed photon light energy. The electron has bounced around to finally reach the primary acceptor, excited to a high energy state, and then it gets passed down an electron pathway that ultimately produces ATP. So once this electron is excited and reaches the primary acceptor, it gets passed on to a molecule called ferredoxin, here shown as FD. And from there, it goes to a series of cytochrome molecules known as the B6F complex. And then along the way, this electron is passed within this complex from acceptor to acceptor, and it releases some of this energy each time, which is used then to phosphorylate. The energy that's released is used to phosphorylate ADP and produce ATP. And once the electron has reached that lower energy states, then it moves back to the photosystem and again back to getting excited and absorbing another photon of light. And in this way, this is a cyclic path in that it once it has gone to its lowest energy state, it's back at the photosystem and it goes through the cycle again. So this process not only produces ATP, but then in order for glucose to be produced, and glucose is another high energy molecule, but ATP is produced, and also another molecule called NADPH. And NADPH is short for nicotinamide adenine dinucleotide phosphate, which is a mouthful. So we just call it NADPH. NADPH is also required in order for glucose to be produced from this cyclic pathway. Now, there's also the non-cyclic electron pathway, and this pathway involves actually both photosystems 1 and 2. And this photosystem is the one that produces ATP and NADPH. So if we were to compare the cyclic pathway, which involves only photosystem 1, this only produces ATP. But we also need the production of NADPH, and NADPH involves the non-cyclic pathway, and it involves both photosystems where ATP and NADPH get produced. So here, photosynthesis begins when a photon of light gets absorbed by photosystem 2 down here, and then the absorption of this photon of light excites the electrons of chlorophyll P680, and the electrons are then captured by the primary acceptor. And this primary acceptor, and then that goes through a series of redox reaction where it gets passed down to the electron acceptor plastoquinone here, called PQ. And because this pathway is non-cyclical, it doesn't go through the same cycle as the cyclic pathway did, then the energized electron will not return back to the photosystem, and therefore it has to be somehow replaced and replenished. So once the electron leaves this photosystem, so once this electron leaves this photosystem and goes to photosystem 1, then there's an enzyme called the Z enzyme that uses energy from light to split a water molecule into hydrogen and oxygen molecules, and through a process called photolysis. The word photolysis means splitting by light. And then the oxygen that is released by that reaction, by the photolysis reaction, is the oxygen that's then released into the atmosphere, the byproduct of photosynthesis. While the electron that is then liberated from this photolytic reaction is then captured by the photosystem, and then that Z enzyme donates the hydrogen ion from that water molecule that went through photolysis. Then that hydrogen molecule, and it donates that hydrogen molecule to this, back to this P680 reaction. So there's that H2O, there's that Z enzyme, and the Z enzyme is carrying out photolysis rather, and releases O2, which escapes into the atmosphere and produces these hydrogen molecules, and these hydrogen molecules are then passed on to the reaction, the P680 reaction center. And these hydrogen ions will join that excited electron and travel with it from electron acceptor to electron acceptor. And that excited electron and hydrogen ion pair move on from plastoquinone to another BF6F complex, the cytochrome complex, and go now towards the P700 complex. And as this electron then moves through that complex, and we are looking now at the movement of that electron through the complex. Here's a closer look at the that non-cyclic pathway where the electron flows from one acceptor to the next. These hydrogen ions that were liberated by photolysis from this Z enzyme, these hydrogen ions then are transported here by that cyclic electron pathway that we talked about above to further increase the hydrogen ion concentration on this side of the membrane. And further, this photolysis reaction that takes place by the enzyme just keeps adding to the hydrogen ion concentration and creating a greater gradient across the membrane. And the movement of these hydrogen ions back across the membrane then releases the energy that then gets used to phosphorylate ADP into ATP. So as these hydrogen ions that were made on this side of the membrane then travel through this concentration gradient, this down their concentration gradient, the energy from this transport is used to phosphorylate ADP into ATP. And this type of ATP formation is referred to as chemiosmosis. So this electron transport chain then produces ATP, produces NADPH that will then feed on into the Calvin cycle. So then between these two electron pathways, you'll have enough ATP made to make enough power for the plant cells, and with enough left over to be sent along with that NADPH towards the Calvin cycle. And if you look at the relative rates of these two cycles, the cyclic and the non-cyclics, you'll see that these are regulated by the amount of NADPH that is available for use. If the levels are very high, cyclic flow will produce ATP, and if the levels are really low, then that will simulate the non-cyclic flow to produce, because the non-cyclic flow does not produce as much. So then in stage 2, those molecules of NADPH and ATP then feed into the Calvin cycle in stage 2, the light-independent reaction. And in this part of photosynthesis, you have ATP molecules and hydrogen ions, which come stored in this NADPH. So there's that hydrogen ions that are stored in NADPH. And these are used then to reduce the carbon that is found in carbon dioxide to ultimately form glucose. And this light independent stage sometimes gets referred to as the dark reaction of photosynthesis, which may sound as if though it is happening in the dark, but it happens both in the dark and in the light. It's just that it's not directly dependent on the presence of light. So that's why it's called the light independent stage, is the better term for it. So yes, this light independent reaction takes place as long as these products of ATP and NADPH are present in sufficient amounts. Then the reactions for the Calvin cycle, for the light independent reactions will proceed. And unlike the light dependent stage, which we talked about, which takes place on photosystems 1 and 2, in the non-cyclic electron pathway and the cyclic electron pathway. So unlike the light independent stage, and those take place on the thylakoid membrane, this stage, this light independent stage, takes place in the stroma of the chloroplast. So here you have, So here you have the thylakoids, and here's the lumen of the thylakoid, and here's the membrane. So this is the site for the light dependent stage, but the light independent stage, the Calvin cycle, takes place in the stroma. So in this space out here, okay? And in this stage, glucose is formed through a series of also cyclic reactions where you have some of the starting material then gets, excuse me, regenerated as the reaction goes on and cycles through. That's why it's referred to as the Calvin cycle. And this Kelvin cycle was named in honor of a scientist called Melvin Kelvin, who was a researcher who determined the steps that were involved in this process. But the cycle itself, we can break down into 3 distinct stages, which we will talk about: carbon fixation, the reduction, and then the regeneration. So if we look at the carbon fixation stage, so first, in order to incorporate carbon molecules into organic molecules, you have to have carbon dioxide brought into the plant through those stomata. And stomata are these openings on the underside of the leaf that allow the exit and entry of carbon dioxide, of oxygen, but also they are very much controlled by the amount of water also that is available. So these stoma allow carbon dioxide to enter the plant cell. And upon entry, it has to be attached to a 5-carbon component called ribulose bisphosphate, which is already present in the stroma of the chloroplast. So if we look at that, this is the first stage, the fixation of carbon dioxide. This represents the entirety of the Kelvin cycle. So the first stage is the fixation of carbon dioxide. So the carbon dioxide is brought in and it gets incorporated into a 5-carbon molecule. So here it is, 1, 2, 3, 4, 5, a 5-carbon molecule, which then becomes, once this one more carbon from the CO2 is added to it, becomes a 6-carbon molecule, but then it's split into 2. So essentially 2 3-carbon molecules. And then we go through the next part of the Kelvin cycle, which is the reduction phase. And then finally, the regeneration of this RUBP. So let's look at those steps a little bit closely. So as I said, first you have this carbon dioxide, which becomes attached to that 4-carbon compound. The 4-carbon compound is, let me just highlight it, is called ribulose bisphosphate, or RUBP for short. And this is a 5-carbon compound, right? So if you add one more carbon from the carbon dioxide to it, you will be resulting in a 6-carbon molecule. And this 6-carbon molecule happens to be very unstable, and it immediately splits into 2 3-carbon molecules of PGA or phosphoglycerate. And this splitting is mediated, this carbon fixation step. This is mediated by an enzyme called Rubisco that catalyzes this reaction. And this part of the cycle has to take place 3 times. Must repeat 3 times before you have enough of the PGA molecule to move into the next stage. So in other words, you need to do this with 3 carbon dioxide molecules, which will use up 3 of this RUBP and will produce 6 of this PGA molecule, or the phosphoglycerate. And so again, 3 carbon dioxide molecules. So 1 carbon each, but 3 of them. So we have 3 carbons. And they will join with 3 RUBP molecules. So 1 carbon dioxide to 1 RUBP molecule. So, but if you have 3 of them, then you need 3 of these. And at this point, this RUBP, since each one of these RUBPs is a 5 carbon molecule, if you have 3 of them, you will have 15 carbons. 15 carbons, add 3 carbons to it, you will have a total of 18 carbons. So you need to make 6 molecules of PGA before these molecules, these 6 molecules, can move on to the next step. So 6 molecules then move on to the next step. But this will only generate enough carbon for half of the glucose molecule. So in order to produce 1 full glucose molecule, you actually need 6 carbon dioxides. So 1 full glucose molecule, 6 carbon molecules need to enter the cycle. Before you can enter the rest. Then the next phase is referred to as the reduction phase, where you have your PGA becomes reduced. So in this phase, you take 6 molecules of PGA. So here you have 6 molecules of phosphoglycerate, or PGA. These 6 molecules become phosphorylated through 6 ATP molecules. So here you can see, showing in this diagram, that these 6 phosphoglycerates become phosphorylated by 6 ATPs. And once the phosphate is transferred to here, you will get 6 ADPs. And this will have, each one of these will have 1 more phosphate attached to it. And this 3-phosphoglycerate will become, so it started out as 3-phosphoglycerate, but because now you've added 1 more phosphate from your ATP, it will be a 1,3-phosphoglycerate, right? So here is that phosphoglycerate molecule with its 3 carbons, carbon 1, carbon 2, carbon 3. And this phosphate from here, from RUBP, is on carbon number 3. So that's why we call this 3-phosphoglycerate. Now, if you add 1 more phosphate onto this molecule, you'll be adding it at the 1, carbon 1. So then here, after this step in the phosphorylation, you will have 1,3-phosphoglycerate, which is where we are here right now. So these ATPs that were used here for the phosphorylation of 3-phosphoglycerate to 1,3-phosphoglycerate, these ATP molecules came from the light-dependent reaction. Next, you need those NADPH molecules to come into play. Those NADPH molecules were also produced by the light-dependent stage, and they need to donate their hydrogen ion and also 2 electrons to this 1,3-phosphoglycerate to reduce it to give 6 molecules of glyceraldehyde 3-phosphate, or GAL. So next come these 6 NADPHs, and they donate their 6 hydrogen ions to here, and also their electrons to produce 6 molecules of, where was I? To produce 6 molecules, here we go, 6 molecules of glyceraldehyde 3-phosphate. And that's the glyceraldehyde 3-phosphate. Another name for it is PGAL, but it's the same thing. Just G3P is the same as PGAL here. And I guess I can actually rename this. Maybe I should rename it so that we can be consistent here with this diagram. Glyceraldehyde 3-phosphate. And same with this one. Let's call it glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So then this NADP plus that gets produced here, then this NADP plus is now free to go back to the thylakoid membrane, and the molecule of G3P then leaves the Calvin cycle. This becomes the product of the Calvin cycle, is this one molecule of glyceraldehyde 3-phosphate. And then the final stage of this Calvin cycle is then the regeneration of RUBP. So once one of these molecules leaves, and remember you had 6 at this point, you had 6 of this glyceraldehyde 3-phosphate, right? You had 6 of them. 1 leaves, that means you've got 5 left behind. So these 5 molecules, and these are 3-carbon molecules, so now you've got 15 carbons. These molecules get rearranged using 3 more ATPs, and they get regenerated to, oops, Ahmed got dropped off. Hi, Ahmed, I'm not sure when we lost you. I hope you didn't miss too much.
Sorry, it was just the Wi-Fi. I'm good now.
Okay.
I only missed like 10 seconds.
Okay, all right, great. So yeah, what we were saying is that at this point in the Calvin cycle, you have 6 molecules of glyceraldehyde 3-phosphate. One of them leaves the Calvin cycle to go towards production of glucose, which means you have 5 more left over. And these 5 more, through the use of 3 more ATP molecules, will go and become regenerated and rearranged to make 3 molecules of RUBP. So if you think of it, the math completely adds up. Here you have 5 3-carbon molecules. And at this point, then you will have 3 5-carbon molecules. And at this point, you have 5 phosphates. And if you add 3 more and rearrange them, the third, these 3 more phosphates will go on to produce this RUBP, which has 2 phosphate molecules on it, 2 phosphate groups on it. And that's why it's called bisphosphate. The BP stands for bisphosphate, which means 2 phosphate molecules, 2 phosphate moieties. So at that point, we are now at the end of the Calvin cycle. and we have produced one glyceraldehyde 3-phosphate, which will then go on to make glucose. So at this point, we're going to now start looking at cellular respiration. But before I do that, I have a really brief video that I can show you. Let me see the time. We have time. So I think maybe I'll continue with respiration, cellular respiration, and see, oh my goodness, I just realized, I think I might be missing out some things. No, it's fine. So this regeneration of this RUBP then allows the whole process of the Calvin cycle to happen all over again. And ultimately, this glyceraldehyde 3-phosphate goes on to be arranged into a molecule of glucose. So now let's look closely at cellular respiration, and then we are going to be able to put the whole connection between cellular respiration and photosynthesis together. So again, just to recap, we have animals that are heterotrophs, that are not able to make their own food, so they are directly dependent on eating plants. And plants are able to generate their own food, so animals that eat plants gain their energy from eating these plants, and they gain it and then they can convert it to a form that they can utilize. So once you have glucose entering the system, there are these cellular processes that are in place to break down that glucose molecule and to release that stored energy, the energy that's stored in the form of the bonds within the atoms of the molecule. And because this process is exergonic, it releases energy, it has to be done in a series of steps in order to control the amount of heat that's produced during this process. If too much heat is allowed to be released all at once, you have a situation that's very similar to combustion. Too much heat and it burns up. But in the cell, you don't want too much heat to be released all at once. So you want to do this through a series of controlled steps that release energy only a little bit at a time, and as a result, don't cause damage to the cell. So when you have this process, there are some single-celled organisms that are able to carry this process out anaerobically or in the absence of oxygen, and this process is referred to as fermentation, and we can look at this a little bit later on in this lesson. But large multicellular organisms have much greater energy needs, and so they really need to release the additional energy that's stored within the glucose molecule in order to survive. So these organisms use the three-part process that we know as cellular respiration in order to fully utilize the energy from a molecule of glucose. So you can think about this a little bit yourself. There is an activity and a thought question. If you Think of holding a clothespin in your hand between your thumb and your index finger of your dominant hand. So if you're right-handed, your right hand. If you're left-handed, your left hand. And then if you were to time yourself into how long it takes for you to quickly open and close this clothespin as completely and as quickly as possible until you can no longer do this. If you were to time this until your fingers are not able to close that clothespin anymore, and then if you were to time it with your other hand, and think about how long will it take for your dominant hand to tire out and get fatigue, how long will it take for your non-dominant hand, do you think, to tire out and get fatigued? How do your fingers feel at the end of such an activity? And why do you think after a while of doing this motion with the clothespin open and closed really as quickly as you can, why do you think you fatigue and your fingers stop working? So these are questions that you can just keep in mind, and we will get back to it further on into the lesson. So I'm just going to pause for a moment to take a drink. Thank you. So to talk about cellular respiration, just think about those energy drinks that are advertised to energize you and to replenish your energy levels when your energy levels are so low. Now, the majority of them are quite highly caffeinated if you read the labels, but if you look closely, you'll also see that they contain even more massive quantities of sugar. And it's this sugar that you're able to break down quickly and produce energy. And this is actually what is driving the energy effects of these energy drinks. And our cells have developed a number of different mechanisms to be able to extract that energy that they need from whatever nutrients are available. And organisms are able to break down those covalent bonds that hold the glucose molecule together through a series of redox type reactions that rearrange that sugar molecule into new and more stable configurations. And during cellular respiration, these redox reactions result in the transfer of electrons from glucose ultimately to oxygen. The glucose is oxidized to carbon dioxide, and the oxygen is reduced to water, hence the redox reaction. And overall, the chemical reaction for cellular respiration is written like this. You have your glucose molecule plus oxygen produces carbon dioxide and water, and also releases energy as heat and in the form of chemical energy stored in the molecule ATP. So again, here in this redox reaction, the glucose is oxidized to carbon dioxide, and the oxygen is reduced to water. And this equation really is just an oversimplification. It's a summary of the process that's taking place. And in reality, this anaerobic portion of the process, when it's combined with aerobic portion, translates to a series of approximately 30 enzyme-controlled steps. And again, these large number of steps is what's necessary in order to dissipate the heat that is released from this reaction. So the body's quote unquote burning of glucose, if you look at this reaction, this is very much similar to a combustion reaction. This burning of glucose is really no different than burning gasoline to run your car or burning propane in your barbecue. When glucose is broken down, this liberated free energy, which is about 2,870 kilojoules per mole of glucose, can be converted to ATP, which then the cell can readily use as its way of powering all of its endergonic processes. And here in this figure, this is an overview of the process of cellular respiration. And you can see that overall, this cellular respiration has got three main stages. The first one being glycolysis, the second one being pyruvate oxidation, and finally another cycle, this time called the Krebs cycle. In glycolysis, you have glycolysis is an anaerobic process. It takes place in the absence of oxygen. Oxygen is not required. In this step, you have a small amount of ATP produced along with the molecule pyruvate. So glucose is converted to pyruvate through glycolysis, and also releases NADPH. And this pyruvate is then shuttled across into the mitochondrial matrix to go through the second stage of cellular respiration, or pyruvate oxidation. Here is where the aerobic portion of glycolysis begins, where this pyruvate molecule is altered and then into acetyl-CoA, and which is then fed into Krebs cycle. And this Krebs cycle then generates ATP, but it also generates molecules of NADH, which together with FADH, which is also produced by the cycle, all feed into the electron transport chain and chemiosmosis here to generate more molecules of ATP through the process of oxidative phosphorylation. And this process, this electron transport, occurs on the membrane-bound proteins that are embedded in the membrane of the mitochondria. So mitochondria is really the organelle of cellular respiration. And unlike photosynthesis, which takes completely within, it's contained within the chloroplast. So if we go back and look at Photosynthesis, it takes place within the chloroplast. Some of it takes place on the membranes of the thylakoid inside the thylakoid, and the rest of it takes place in the stroma of the chloroplast, but it's entirely contained within the chloroplast. But in contrast to photosynthesis, cellular respirations happens partially within the cytoplasm. So out here, if you can imagine, this is the mitochondria, which means out here is the cytoplasm and the rest of the cell. Again, if we look at this image of the plant, so here is a mitochondria. Here is a chloroplast. So photosynthesis takes place entirely within here, but cellular respiration, some of it takes place out here within the cytoplasm, and some of it takes place on the mitochondria. So the mitochondria is this oval-like structure, and it's composed again of two membranes, an outer membrane and an inner membrane, which is actually quite highly folded. The inner membrane is very highly folded, and the inner folded portions of the membrane are referred to as cristae. And the reason they're folded is to create even greater surface area to allow even more proteins and enzymes that are associated with the final stages of cellular respiration to be present and crammed into this into the membrane of the mitochondria. So as well, this the other thing that this inner membrane does, it in effect creates two compartments within the mitochondria. The innermost part of the mitochondria, or the mitochondrial matrix, contains a very rich, protein-rich type of liquid. And then there's the intermembrane space between the inner and the outer membrane. And the intermembrane space is also fluid-filled. And both of these compartments, both the intermembrane space and also the mitochondrial matrix, the inner part of the mitochondria, both of them play an important role in cellular respiration. So as we talked about, there are several stages to glycolysis. Several stages to cellular respiration. Stage one is glycolysis. It's called glycolysis. And in this stage, this is the part that takes place within the cytoplasm. So here's the cell. Glycolysis takes place out here within the cytoplasm, not within the mitochondria. And this is an anaerobic process, meaning that oxygen is not required by this stage, by glycolysis. And in this stage, the goal of glycolysis is to take the 6-carbon glucose molecule and ultimately split it into 2 3-carbon molecules of pyruvate. So you start with the 6-carbon glucose, and by the end of glycolysis, you will have 2 pyruvate molecules, which each of them have 3 carbons. And if you actually look at the term glycolysis, it literally means sugar splitting. Glyco, sugar, and lysis means breaking or splitting. In glycolysis, although we can just as a summary, just think of it as 6 carbons for glucose going in to produce 2 pyruvates that are 3 carbon each. but it actually takes place in 10 distinct steps. And each one of these steps is catalyzed by a different enzyme. You can think of the first 5 steps are referred to as the energy investment steps. So if you look at steps 1, 2, 3, 4, and 5, these are the energy investment steps, where the cell uses up ATP. So if you look, you see ATP is being fed in. Whereas and if you think of the consumption, it's minus 1 ATP, meaning that 1 ATP was used and another ATP was used. But the next, the last 5 steps are the energy payoff phase of glycolysis, where then ATP is produced. You can see ATP is produced here in these final stages through a process called substrate level phosphorylation. And in this type of ATP production, ATP is formed directly when you have a phosphate containing compound transferring a phosphate to ADP to form ATP. This is called substrate level phosphorylation. So in the first step, and I've tried to lay it out here so that these steps kind of align with the steps here in the reaction pathway. The first step is where you have ATP used up to donate its phosphate group and transfer it to carbon 6 of the glucose molecule to give glucose 6 phosphate. That means you have glucose, and at position 6 on its 6th carbon, you have now positioned a phosphate. So glucose goes to glucose 6 phosphate. In the second step, this molecule of glucose 6 phosphate is then rearranged or isomerized to form fructose 6 phosphate. In the third step, another ATP gets added in, transferring its phosphate this time to carbon 1 of the fructose molecule, now making it fructose 1,6 bisphosphate. This means there's a molecule of fructose with 2 phosphates, in other words, bisphosphate, 2 phosphates. Where are these phosphates? At carbon 1 and carbon 6. So fructose 1,6 bisphosphate is then split into 2 different molecules. It's split into a molecule of glyceraldehyde 3-phosphate and 1 molecule of dihydroxyacetone phosphate, or DHAP. So G3P and DHAP. This molecule of fructose 1,6 bisphosphate is split into 1 of this and 1 of this. Now, in the 5th step, and really the 5th step is just here, this is just a continuation. I guess the best thing to do is for this arrow just to carry on straight down. This is the 5th step. In the 5th step, this dihydroxyacetone phosphate, which is actually an isomer of this glyceraldehyde 3-phosphate, becomes rearranged to produce glyceraldehyde 3-phosphate. So at the end of the 5th step, you actually have 2 molecules of glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is then what continues through the rest of the reaction. In steps 6 to 10, you have 2 molecules of glyceraldehyde 3-phosphate continuing through the rest of the reaction path, effectively doubling the product that's created at the end. So if you can think of whatever is happening in 1 with one of these molecules is also happening with the other molecule. So every time you produce an NADH, you're actually producing two. Every time you're producing an ATP, you're actually producing two, because it's happening with two molecules of glyceraldehyde 3-phosphate. In step 6, you have your glyceraldehyde 3-phosphate being oxidized by the transfer of electrons and protons to the carrier molecule NAD+ to create NADH. And also an inorganic phosphate is also added at this step, creating 1,3-bis-phosphoglycerate. And because this process is an exergonic process, the energy that gets released from this process is actually used up for the attachment of this inorganic phosphate from the cytoplasmic medium to carbon 1 of glyceraldehyde 3-phosphate. And so now you have not only a phosphate at carbon 3, but also a phosphate at carbon 1. So that's why this molecule is named 1,3-bis, meaning 2, bis-phosphoglycerate. 2 phosphate groups at carbon 1 and carbon 3 on a glycerate molecule. So in other words, now we have attached 2 inorganic phosphates and have 2 molecules of 1,3-bis-phosphoglycerate and have made 2 molecules of NADH. In step 7, which would be this one right here, 1,3-bis-phosphoglycerate going into 3-phosphoglycerate, you get what we talked about as substrate level phosphorylation, which is used to transfer the phosphate directly from this substrate molecule. That's why it's called substrate level phosphorylation, directly from the substrate molecule to a molecule of ADP, therefore making it an ATP. That phosphate comes from carbon number 1. So now that you have transferred it to the ADP making ATP, the resulting molecule is just 3-phosphoglycerate. It's lost one of its phosphate from carbon number 1. And just keep in mind, this is happening in both of these branches. So in effect, you are creating 2 molecules of ADP, shown right here. And right. So then in step 8, you have another transfer of phosphate to ATP. ATP, this remaining phosphate group. First you have a rearrangement. The phosphate group that was on carbon number 3 gets rearranged and put onto carbon number 2. So 3-phosphoglycerate becomes 2-phosphoglycerate. That phosphate got moved on to carbon 2. And then you have step 9, where a molecule of water gets removed. And by doing so, this removal of molecule of water creates a double bond and leads to the rearrangement of the electrons. And this rearrangement of electrons creates a molecule that is highly unstable. And the phosphate group on it is highly labile. And so as a result, that phosphate group on the PEP molecule goes through another substrate level phosphorylation again, generating another ATP. In reality, 2 more ATPs at this step of the glycolytic pathway, 2 more ATPs, and leaving behind a molecule of pyruvate at the end. So at the end of these 10 steps that we talked about here, in this first anaerobic stage of cellular respiration and glycolysis, at the end of glycolysis, the end of these 10 steps, you have 2 molecules of ATP made, 2 molecules of NADH, and 2 pyruvate molecules. So in net total, you put in 2 molecules here, you made 2 molecules here, so these 2 can cancel out. So in net, you've made 2 ATPs and 2 NADHs and 2 pyruvates at the end of glycolysis. So I'm just going to pause here, and I'm going to spend the next few minutes just, we can watch a video together that goes through the steps of photosynthesis, and hopefully we'll show in a more animated way all of these steps. I'm just going to bring this over here. So this is one of these crash course videos that features this guy here, and he talks fast and cracks a lot of jokes. Hope you find it entertaining. Altogether, I think this video, is 13 minutes. I think we have enough time to watch it.
Photosynthesis. It is not some kind of abstract scientific thing. You would be dead without plants and their magical, nay, scientific ability to convert sunlight, carbon dioxide, and water into glucose and pure, delicious oxygen. This happens exclusively through photosynthesis, a process that was developed 450 million years ago and actually rather sucks. It's complicated, inefficient, and confusing, but you are committed to having a better, deeper understanding of our world, or more probably you'd like to do well on your test. So let's delve. There are 2 sorts of reactions in photosynthesis, the light-dependent reactions and the light-independent reactions, and you've probably already figured out the difference between those 2, so that's nice. The light-independent reactions are called the Calvin cycle. Yes, that Calvin cycle. Photosynthesis is basically respiration in reverse, and we've already covered respiration, so maybe you should just go watch that video backwards. Or you could keep watching this one. Either way, I've already talked about what photosynthesis needs in order to work: water, carbon dioxide, and sunlight. So how do they get those things? First, water. Let's assume that we're talking about a vascular plant here. That's the kind of plant that has pipe-like tissues that conduct water, minerals, and other materials to different parts of the plant. These are like trees and grasses and flowering plants. In this case, the roots of the plants absorb the water and bring it to the leaves through tissues called xylem. Carbon dioxide gets in and oxygen gets out through tiny pores in the leaves called stomata. It's actually surprisingly important that plants keep oxygen levels low inside of their leaves for reasons that we will get into later. And finally, individual photons from the sun are absorbed in the plant by a pigment called chlorophyll. All right, you remember plant cells? If not, you could go watch the video where we spend the whole time talking about plant cells. One thing that plant cells have that animal cells don't: plastids. And what is the most important plastid? The chloroplast, which is not, as it is sometimes portrayed, just a big fat sack of chlorophyll. It's got complicated internal structure. Now, the chlorophyll is stashed in membranous sacs called thylakoids, and the thylakoids are stacked into grana. Inside the thylakoid is the lumen, and outside of the thylakoid, but still inside of the chloroplast, is the stroma. The thylakoid membranes are phospholipid bilayers, which, if you remember, means that they are really good at maintaining concentration gradients of ions and proteins and other things. This means keeping a concentration higher on one side than the other of the membrane. You're going to need to know all of these things. Sorry. Now that we've taken our little tour of the chloroplast, It's time to get down to the actual chemistry. First thing that happens, a photon created by the fusion reactions of our sun is about to end its 93 million mile journey by slapping into a molecule of chlorophyll. This kicks off stage one, the light-dependent reactions, proving that yes, nearly all life on our planet is fusion powered. When chlorophyll gets hit by that photon, an electron absorbs that energy and gets excited. This is the technical term for electrons gaining energy and not having anywhere to put it. And when it's done by a photon, it's called photoexcitation. So let's just imagine, for the moment anyway, that every photon is whatever dreamy young man 12-year-old girls are currently obsessed with, and electrons are 12-year-old girls. The trick now, and the entire trick of photosynthesis, is to convert the energy of those electrons into something that the plant can use. We are literally going to be spending the entire rest of the video talking about that. I hope that that's okay with you. That first chlorophyll is not on its own here. It's part of an insanely complicated complex of proteins and lipids and other molecules called photosystem 2 that contains at least 99 different chemicals, including over 30 individual chlorophyll molecules. This is the first of 4 protein complexes that plants need for the light-dependent reactions. And if you think it's complicated that we call the first complex photosystem 2 instead of photosystem 1, then you're welcome to call it by its full name, which is plastoquinone oxoreductase. Oh, no, you don't want to call it that? Right then, photosystem 2. Or, if you want to be brief, PS2. PS2 and indeed all of the protein complexes in the light-dependent reactions straddle the membrane of the thylakoids in the chloroplasts. Now that excited electron is going to go on a journey designed to extract all of its new energy and convert that energy into useful stuff. This is called the electron transport chain, in which energized electrons lose their energy in a series of reactions that capture the energy necessary to keep life living. So PS2's chlorophyll now has this electron that is so excited that when a special protein designed specifically for stealing electrons shows up, the electron actually leaps off of the chlorophyll molecule onto the protein, which we call a mobile electron carrier because it's a mobile electron carrier. Chlorophyll then freaks out like a mother who has just had her 12-year-old daughter abducted by a teen idol and was like, what do I do to fix this problem? And then it, in cooperation with the rest of photosystem 2, does something so amazing and important that I can barely believe that it keeps happening every day. It splits that ultra-stable molecule, H2O, stealing one of its electrons to replenish the one it lost. The byproducts of this water splitting, hydrogen ions, which are just single protons, and oxygen. Sweet oxygen. This reaction, my friends, is the reason that we can breathe. Brief interjection. Next time someone says that they don't like it when there are chemicals in their food, please remind them that all life is made of chemical feels. When you say that, it spends all of its time and energy creating the air we breathe, and we're like, ew, chemicals are so gross. Now remember, all energized electrons from PS2 have been picked up by electron carriers and now are being transported to the second protein complex, the cytochrome complex. This little guy does 2 things. 1, it serves as an intermediary between PS2 and PS1, and 2, it uses a little bit of that energy from the electron to pump another proton into the thylakoid. So the thylakoid is starting to fill up with protons. We've created some by splitting water, and we moved one end using the cytochrome complex. But why are we doing this? Well, basically what we're doing is charging the thylakoid like a battery. By pumping the thylakoid full of protons, we're creating a concentration gradient. The protons then naturally want to get the heck away from each other, and so they push their way through an enzyme straddling the thylakoid membrane called ATP synthase. And that enzyme uses that energy to pack an inorganic phosphate onto ADP, making ATP the big daddy of cellular energy. All of this moving along the electron transport chain requires energy, and as you might expect, electrons are entering lower and lower energy states as we move along. This makes sense when you think about it. It's been a long while since those photons zapped us and pumping hydrogen ions to create ATP. It's splitting water and jumping onto different molecules. Tired just talking about it. Luckily, as 450 million years of evolution would have it, our electron is now about to get re-energized upon delivery to photosystem 1. So PS1 is a similar mix of proteins and chlorophyll molecules that we saw in PS2, but with some different products. After a couple of photons re-excite a couple of electrons, the electrons pop off and hitch a ride onto another electron carrier. This time, All of that energy will be used to help make NADPH, which, like ATP, exists solely to carry energy around. Here, yet another enzyme helps combine 2 electrons and 1 hydrogen ion with a little something called NADP+. As you may recall from our recent talk about respiration, there are these sort of distant cousins of B vitamins that are crucial to energy conversion. In photosynthesis, it's NADP+, and when it takes on those 2 electrons and 1 hydrogen ion, it becomes NADPH. So what we're left with now after the light-dependent reactions is chemical energy in the form of ATPs and NADPHs. And also, of course, we should not forget the most useful, useless byproduct in the history of useless byproducts, oxygen. If anybody needs a potty break, now would be a good time, or if you want to go rewatch that rather long and complicated bit about light-dependent reactions, go ahead and do that. It's not simple, and it's not going to get any simpler from here. Because now we are moving along to the Calvin cycle. The Calvin cycle is sometimes called the dark reactions, which is kind of a misnomer because they generally don't occur in the dark, they occur in the day along with the rest of the reactions. But they don't require energy from photons, so it's more proper to say light-independent reactions. Or if you're feeling non-descriptive, just say stage 2. Stage 2 is all about using the energy from those ATPs and NADPHs that we created in stage 1 to produce something that's actually really useful for the plant. The Calvin cycle begins in the stroma, or the empty space inside of the chloroplast, if you remember correctly. And this phase is called carbon fixation because, yeah, we're about to fix a CO2 molecule onto our starting point, ribulose bisphosphate, or RUBP, which is always around in the chloroplast because not only is it the starting point of the Calvin cycle, it's also the end point. That's why it's a cycle. CO2 is fixed to RUBP with the help of an enzyme called ribulose 1,5-bisphosphate carboxylase oxidase, which we generally shorten to Rubisco. I'm in the chair again. Excellent. This time for a biography of Rubisco. Once upon a time, a one-celled organism was like, man, I need more carbon so I can make more little me's so I can take over the whole world. Luckily for that little organism, there was a lot of CO2 in the atmosphere, and so it evolved an enzyme that could suck up that CO2 and convert inorganic carbon into organic carbon. This enzyme was called Rubisco, and it wasn't particularly good at its job, but it was a heck of a lot better than just hoping to run into some chemically formed organic carbon. So the organism just made a ton of it to make up for how bad it was. Not only did the little plant stick with it, it took over the entire planet, rapidly becoming the dominant form of life. Slowly, through other reactions known as the light-dependent reactions, plants increased the amount of oxygen in the atmosphere. Rubisco, having been designed in a world with tiny amounts of oxygen in the atmosphere, started getting confused. As often as half of the time, Rubisco started slicing ribulose bisphosphate with oxygen instead of CO2, creating a toxic byproduct that plants had to deal with in creative and specialized ways. This byproduct, called phosphoglycolates, believed to tinker with some enzyme functions, including some involved in the Calvin cycle. So plants have to make other enzymes that break it down into an amino acid, glycine, and some compounds that are actually useful to the Calvin cycle. But plants had already sort of gone all in on the Rubisco strategy, and to this day, they have to produce huge amounts of it. Scientists estimate that at any given time, there are about 40 billion tons of Rubisco on the planet. And plants just deal with that toxic byproduct. Another example, my friends, of unintelligent design. Back to the cycle. So ribulose bisphosphate gets a CO2 slammed onto it, and then immediately the whole thing gets crazy unstable. The only way to regain stability is for this new 6-carbon chain to break apart, creating 2 molecules of 3-phosphoglycerate. And these are the first stable products of the Calvin cycle. For reasons that will become clear in a moment, we're actually going to do this to 3 molecules of RUBP. Now we enter the second phase, reduction. Here we need some energy, so some ATP slams the phosphate group onto the 3-phosphoglycerate, and then NADPH pops some electrons on, and voila, we have 2 molecules of glyceraldehyde 3-phosphate, or G3P. This is a high-energy 3-carbon compound that plants can convert into pretty much any carbohydrate, like glucose for short-term energy storage, cellulose for structure, starch for long-term storage. And because of this, G3P is considered the ultimate product of photosynthesis. However, unfortunately, this is not the end. We need 5 G3Ps to regenerate the 3 RUBPs that we started with. We also need 9 molecules of ATP and 6 molecules of an ADPH. So with all these chemical reactions, all of this chemical energy, we can convert 3 RUBPs into 6 G3Ps, but only one of those G3Ps gets to leave the cycle. The other G3Ps, of course, being needed to regenerate the original 3 ribulose bisphosphates. That regeneration is the last phase of the Calvin cycle. And that is how plants turn sunlight, water, and carbon dioxide into every living thing you've ever talked to, played with, climbed on, loved, hated, or eaten. Not bad, plants. And I hope you understand. If you don't, not only do we have some selected references below that you can check out, but of course, you can go rewatch anything that you didn't get. And hopefully, upon review, will make a little bit more sense. Thank you for watching. If you have questions, please leave them down in the comments below.
Right. So we're going to leave off here today, having reviewed photosynthesis from our last lesson, and having started the first part of cellular respiration. And we'll stop here at the end of the first stage, which is glycolysis. So we're going to stop right here. And next class, we are going to continue with the rest of this and also talk about how cellular respiration and photosynthesis are intricately tied together and complementary to one another. Anybody have any questions? All right, everyone. In that case, I think this is a good place to end our lesson.